welcome to this introductory video for the series about the profile aligner. I actually recorded this video after completing the subsequent ones because I realized that covering some foundational concepts would make the series more accessible. So treat this video as a prequel and we'll be covering 3 essential topics. First, I'd like to introduce the basic problem we are trying to solve. And for this, we need to understand what sequence alignment algorithms are. Second, I will provide the biological context about the cis-regulatory elements in the genome. Lastly, I'll talk about the computational background, specifically covering dynamic programming approaches and memoization matrices in the context of sequence alignment. If you're already comfortable with these concepts, feel free to skip to the next videos. So, the profile aligner is a sequence alignment algorithm. But what does that even mean? The word sequence here is referring to nucleotide or amino acid sequences. We are probably familiar with nucleotides that we denote as A, C, G, and T, and different arrangements of these nucleotides make a DNA sequence. The word alignment, we can understand it as a comparison of 2 or more sequences. For example, when geneticists search for a mutation responsible for a certain disease, they will compare, or in other words, align the DNA of a healthy and diseased individual. And over here, we have an example of an alignment of a short sequence. So let's say the sequence on top is the original sequence. And I've highlighted the mutations that happened to get from the original to the mutated sequence. We see that there was a substitution mutation that converted C to T. We see that there was a deletion of these 2 nucleotides, insertion of these 2, and another substitution from T and G, T to G. By looking at this alignment, we were able to infer mutation events that took us from the original sequence to the mutated sequence. However, there is no historical record that tells us exactly which mutations happened during evolution. And this is where the algorithm part comes in. We rely on alignment algorithms to computationally determine the most likely mutations that took place. So, to summarize, the sequence alignment algorithms allow biologists to computationally reconstruct evolutionary histories and understand genetic relationships. The next item on our agenda is to understand the cis-regulatory elements. Our DNA consists of the coding and non-coding regions. The coding regions are responsible for encoding the proteins, and they are highly conserved because mutations in this region can alter the protein structure and be harmful for the organism. The non-coding regions, on the other hand, were once dismissed as junk DNA, but actually, there are 2 categories. There are neutral regions with no known function, and then there are functionally important regulatory elements, highlighted in black over here. These highlighted parts are supposed to represent the motifs. Motifs are short recurring fragments where regulatory proteins known as transcription factors bind. So these are the transcription factors. And these regulatory elements act as molecular switches that control where, when, and how much of a gene is expressed. We know that non-coding regions show more variability across species compared to the coding regions. This happens because many non-coding segments contain neutral sequences where mutations have few consequences for the organism. And this brings me to the underlying problem the profile aligner attempts to solve. The traditional alignment algorithms were designed primarily for the coding sequences, which have high conservation, as we already mentioned. When we apply those algorithms to more variable non-coding regions, the algorithms become less reliable. the profile aligner attempts to mitigate this issue by integrating information about individual base functional importance. But we'll get to this part in later videos. Finally, let's talk about dynamic programming and memoization matrices, the computational foundation of the profile aligner. You can find plenty of resources on this topic, but I wanted to introduce it in the context of sequence alignment. Dynamic programming is a problem-solving approach that breaks complex problems into smaller, overlapping subproblems. But what does that mean in the context of sequence alignment? To understand, we can consider this problem or a goal where we are trying to align sequences ACT and AGT. So I've represented an initial breakdown of the problem, the root node representing the overarching one. So here I've placed these two sequences in a function of f. This f can be any optimization function that you're using. And the way we create smaller subproblems from here is by chopping off the last bases from the original sequence. And there are three ways we can chop it off. First, we can remove a base from either the top or the bottom sequence, or we can remove both of the last bases, which is over here. And then we go on and break down these subproblems into even smaller subproblems. For example, if we look at this middle node, we see that I've broken it down by removing the last element of the top part or the last element of the second sequence. And if we take a look here to this subproblem, we see that they are the same. And this brings me to the core principle of dynamic programming, where we have overlapping subproblems and we solve each of them only once and store their solution in order to avoid redundant calculations. This is particularly important for sequence alignment because the number of possible alignments grows exponentially with sequence length. I mentioned storing the solutions of the overlapping subproblems, and that's where we use the memoization matrices. So, for example, we had a subproblem with aligning AC and AG highlighted in yellow. We can find the corresponding cell for this subproblem, which is over here. We would store the solution to this problem in this particular cell. And once we would fill out all this entire matrix, we would trace it back to reconstruct the alignment. This is the essence of dynamic programming and sequence alignment. And now we've covered alignment algorithms, cis-regulatory sequences, and dynamic programming, and we are ready to dive deeper. See you in the next videos.